everyone. Welcome to episode 5 of Interview with Scientists series. Today we have Dr. Sanjib Guha from the United States of America. He works at University of Rochester, upstate New York. Dr. Guha was born and raised in India, where he did his undergraduation in 2008, and then he moved to California to complete his master's. Post that, he moved to Singapore to work as a research associate at National University of Singapore. And then he moved to Barcelona, quite an interesting city, to do his PhD. And after completion of that, he again moved back to California, where he did his first postdoc at the Buck Institute. And now he's doing his second postdoc at University of Rochester, New York. Welcome, Dr. Guha.
Thank you for having me.
You're most welcome. So tell us about your research career. What motivated you to become a scientist, and what are your plans after completing your postdoc?
Yeah, that's a long story. So to start back, my grandfather, he was a cardiac surgeon.
Yes.
And I used to hear stories from my grandmom. It was around independence era, 1950s, I would say. He used to travel all over India. Of course, we just had Pakistan and maybe
Yes.
Bangladesh following that. So I used to hear from my grandma that he used to travel all over India and maybe sometimes across the borders, to treat the patients and everything. And he used to, most of the time, for free, all the social activities he used to do.
Yes.
So obviously I could not meet him in person, but I used to hear all these stories from my grandmom, and that motivated me a lot, something I need to do for the community as well. And of course, it's a very difficult.
Yeah.
But unfortunately, becoming a doctor is so difficult in India. It's so much competition.
Yes.
And that happened with me. And so then I decided, why not pursue biology, which is also another very interesting career field and very related to medical field, especially the kind of work we always do. And with that, as you said, I did my undergrad studies in 𝐬𝐜𝐢𝐻 Institute in Kolkata, biotechnology as my BTech. And after that, I decided to follow foreign dreams, because my close uncle, my dad's brother, he was in US for a long time. He's still working in the US Army.
Yeah.
So I thought maybe it will be good for me to move to US as well. And I gave those competitive exams, GRE, TOEFL and everything. And then I moved in 2008 when I was hardly 22, 23 years old, and came to a foreign land to pursue my master's. And then obviously I fall in love with the subject, and then gradually things happen. I did my PhD, first postdoc, second postdoc, and now coming back to the question, what's next? So already I have been offered from my PIs to be assistant professor position.
Okay.
But as you know, there's some visa things that we have to take care of it first to accept that position. So currently I'm working on that, trying to get my H1B done as soon as possible. So if I get the visa, then definitely I will move into assistant professor position in the Rochester University.
I see. That's interesting. So you always wanted to be an academic, a professor?
Well, as of now, yes, but definitely at some point in my career, I like to switch into industry position as well, just to see how things are over there. If it works out well, then I can pursue that. But if it doesn't work out, I can anytime come back to academics. But definitely at some point, just to see, explore other options. How the situation over there. And as I said, if it doesn't work well, I can anytime come back to the academic environment.
Nice. So I read your papers. You work with Alzheimer's disease. So can you tell us briefly what is Alzheimer's disease and how do you model it in animals?
Yeah, so Alzheimer's disease is one of the major neurological disorders affecting millions of people worldwide, and is also the most common form of dementia.
Yes.
And the bad thing is it happens for people over 60 years of age. It's called age disease, and I think there is a ratio of 1 to 5 people getting affected with this disease, especially in the current scenario. So it's a very destructive disease, and over time, people lose their memory, they lose their cognitive functions, and starting gradually all the functions that are controlled by brain and central nervous system gradually loses out.
Yes.
And unfortunately, again, like there are no therapeutic treatment options available. Like whatever top counter medicines are there, like it will not like cure the disease. Maybe it can like stop the progression of the disease. Like people cannot go even worse, but it will not bring them back to the normal healthy level. So that's a very, very serious disease. And obviously, in our lab scenario, like we use different model organisms. I mainly work with C. elegans,
Yeah.
but we also have mice setup in the lab as well. So after this initial paper is out, like we will definitely transition into higher model organisms.
Yes.
So what we do in our case, like in Alzheimer's disease, like there are two main pathological hallmarks. Like one is A beta protein and other one is tau protein. So we, with the help of microinjection, we specifically integrate this tau protein into the C. elegans genome. And our main purpose is to see, like when we have this toxic protein inside the system, what are the pathological phenotypes it causes to those tiny little worms.
Okay.
And that's how we try to model. Obviously, there is a huge, like people can say, like there is a huge gap between the model system and as well the human brain, but it's like at a basic lab scenario, we cannot directly work with the human patient samples, right? So we have to start
Yes.
from the very basic and then we gradually progress upward. So
Okay.
that's why we try to see what are the phenotypes that have been caused with the introduction of this toxic protein, and then
Yes.
what are the treatment options we can avail, and then obviously we can move into higher model organisms.
I see. That's interesting. So you just mentioned about tau. What is tau? What's the importance of tau protein
Yeah,
and
yeah, that's a very good question. So tau is a very, in normal physiological conditions, tau is a very important protein. Like all of us have tau. It's a neuronal protein and it helps to conduct like variety of neuronal functions. So it's, first of all, it helps in stabilization of microtubules, which is the backbone of the neuronal integrity and structure.
yes.
And due to this, it helps in like different neural functions such as synaptic transport, synaptic transmission across the neurons. So it is a very beneficial protein.
Okay.
But the problem happens like when it starts from aggregation or when it gets modified due to certain post-translation modification which alters its structure and function. Then
Okay.
due to this modified tau protein, then it starts causing different pathological phenotypes. And then it becomes toxic and started to form aggregates which form lumps like
Okay.
structure between the neurons, and it blocks synaptic connection. And that's how all the neuronal circuit gets damaged gradually. And over time, since these aggregates cannot be removed from the system, they get bigger and bigger. And in this way, like the whole brain system gradually loses its function. And that's
Okay.
how they get significantly shrink compared to the healthy human brain, the Alzheimer patient brains. And also their other cognitive declines starts to happening. Their, as I say, like their memory starts to fade, and gradually, like over time, it causes variety of other toxic
Okay.
functions.
So we can say that tau is a good thing to have, but once it goes bad, then
Yeah,
we
yeah,
can then it can lead to Alzheimer's, right?
exactly. So as I said, like all of us have tau. So in
Yes.
normal scenario, it carries out various good functions, but as soon as it gets modified,
Yeah.
be it genetic, be it like post-translation modifications or anything, it starts to cause different toxic functions.
Okay. And what's the impact of tau on mitochondria?
Yeah, so that is the cool link that we're trying to find out, because since we are talking about Alzheimer's disease, like which is like a neuronal disease, and the
Yes.
main thing we are thinking about the backbone of neuron. And when we go deep inside the neuronal cell, although we have different cellular organelles, but the key organelle that comes in our mind is mitochondria.
Yeah.
Because from our high school studies, we have learned like mitochondria is the powerhouse of the cell. It generates all the ATP, which helps the neuron to carry out different functions. So that's why we thought like maybe there can be a connection between the tau and the neurons and the neural mitochondria
Mhm.
specifically. And that's what we have found out in our first paper. Like when we have this modified tau protein, not the wild type tau protein, when we have this modified tau protein, it causes different mitochondrial abnormalities, such as their morphology gets altered, their trafficking across the neuron, which is a very cool experiment that we have done in the live confocal imaging. We see
Yeah.
the mitochondria needs to be trafficked along the neuron. But we have seen like when we have this modified tau protein, it actually causes a barrier, like a road blockade. So imagine like if you're driving in the highway and there are like big blocks of like boulders putting in the
Yes.
highway, and the cars gradually stops, right? And
Yes.
the normal passage or smoothness gets disrupted. So
Yeah.
same thing happens in the neuron when we have this modified big aggregate
Yeah.
tau protein in the neuron. It basically blocks the transport of mitochondria, and that's why
Okay.
they cannot get trafficked normally, and which causes like synaptic transmission loss, eventually ultimately causing neuronal structural integrity and everything. And
I
also
see.
we have seen like their fission and fusion, which is part of their morphology, that also gets disrupted. And also the mitophagy, which is also another dynamic mechanism where all the bad mitochondria needs to be removed. That
Yeah.
also gets significantly altered when we have modified tau protein. So this is all about in my first paper, which has been recently accepted in molecular neurodegeneration. And we
Yes.
have, like, I think maybe in the model system specifically, we are the first group which has shown that when we have this modified tau protein, it causes an effect in the mitochondria. Like other people have shown, like maybe there can be a neurodegeneration or any other effects, but
Mm-hmm.
there can be a cool link between mitochondria and the toxic tau. That is probably we are the first group to show that specifically in the model organism.
That's interesting. So, in your recent papers, and you mentioned that in your research, you use C. elegans. So, why did you decide to use C. elegans, not mice models or higher model organisms like chimpanzees?
Yeah,
And
so
how is it different from mice models?
yeah, C. elegans is a very good model organisms because their lifespan hardly lasts for four weeks. So as we are working with a progressive neurodegenerative disorder, so we wanted to model or get a model organism where we can do the experiments or the assays like very quickly, very rapidly, and at different stages of their life cycle. So
Yeah.
we can, for example, the whole, like for example, this trafficking experiment, right? Whole experiment we can conduct in the larval stages. Then we can age them like 7 days, 7-day old C. elegans, and then we can age them further seven days, and then we can do in the 14 days life cycle. So this kind of assays we can do along with their ages, and we can see with the age whether there is a decline in any of these assays or not. But
Okay.
if we have to use mice model, of course we will use it in future. We have to wait like at least 8 to 10 months to do one single experiment.
Okay.
Whereas with this tiny small model organism, we can conduct the whole assay within 4 weeks. So that is also one of the major reason we chose C. elegans, because since, as I said, like since we are working with a progressive disease, we wanted to say like see like whether the pathogenic effect that we are seeing in their developmental stages, whether it further exacerbates when they age in the older worms. So
Okay.
that's one of the main reason. And the other reason is like C. elegans has a very transparent body.
Uh-huh.
So a lot of these experiments that we do, we introduce any fluorescent protein inside the worm genome. So we need to do microscopy assays, and since they have like the transparent body, all this fluorescent protein can be seen live in their body itself.
Okay.
So we don't need to kill the worms, or we don't need to dissect the worms. We can directly put the live worms under a microscopic slide and just take images and videos. Like for example, this trafficking example experiment, we used a protein called 𝐬𝐜𝐢𝐬, which is specifically targeted to the mitochondrial matrix. So
Okay.
just putting the worm in a high resolution microscope, you can see the movement of the mitochondria across the neuron. So
Okay.
that is one of the very big advantage of this model organism.
Okay. So, can you tell us about the treatment options for Alzheimer's that are currently available? And are there any medications or therapy for treating Alzheimer's?
Yeah, so this has been a major problem in this Alzheimer's field. Like a lot of research has been going on. A lot of them went to the third stage clinical trial, but none of them were successful. Like there are a couple of over-the-counter medications as are available, as I said very early in
Yeah.
our conversation. Like they will not stop the Alzheimer's disease, but they will not like bring back to your normal level. But maybe they can alter the progression of the disease. So those
I,
medications are available, but there's very cool work going on. Like for example, I know a company called Aravinus. I think they're in Oregon, most likely.
okay.
They have developed a tiny molecule called PROTAC, PROTSC. So what they do, like they introduce that molecule inside the body, for example, and they directly bind with the modified tau, and
Yeah.
then they capture that tau and move it to directly to the proteasome to degrade them. So that I think is a very cool thing, because if you have this toxic tau in the system, they need to be degraded. Otherwise,
Yes.
as I said, like they will just bind and bind and make like a clump or aggregates. So this
Yeah.
molecule actually directly specifically bind with those tau and target towards the proteasome and eventually
Okay.
get degraded. So I think that one of the very promising thing that is going on. And also there are currently certain immunotherapies available as well, like there are certain antibodies which are specifically targeting the pathological epitopes or sites of tau, directly binding with those phosphorylation or acetylation sites,
Okay.
and also blocking those kind of modification.
Yeah.
So those options are currently available as well. I think they are in phase 3 trial or not. And if you ask me what we are trying to work out with our studies,
Mhm.
since we have certainly found out mitochondrial connection, so
Yes.
in future, of course, what we will like to do, we will try to target not just the tau, we will try to target the mitochondria as well. So we are trying to find out any kind of combination of two drugs, for example, which can block the tau progression or tau aggregation and
Okay.
simultaneously boost up mitochondrial health. So it will be like a dual approach. So in that way, like your pathogenic tau is degraded and
Yes.
simultaneously your mitochondrial health is also being increased further as well.
Okay.
So yeah, so coming back to your previous question, like being using 𝐬𝐜𝐿, I think 𝐬𝐿 is a great model system to do any kind of drug screening. So I
Yes.
think next year we are in line to do a huge drug screening with this, which will simultaneously do this dual kind of functions, and then we can do like a cocktail approach and see which optimum dose works better for killing, like killing a bird with two stones. So
Yeah,
we are quite optimistic about that.
that's very interesting. So
Yeah.
why does this happen? Like some people develop Alzheimer's at late stages, like a 70 or 80 years, but some don't. What?
Yeah. Yeah, so it depends upon lot of things. It's not like just one single factor. Like obviously, I can say genetic, but if you rule out the genetic aspect, it's like the way you carry out your lifestyle. It's like sometimes unhealthy diet, food is one of the major reason. Sometimes like people are more exposed to toxic environment, like working in coal mines or anything like that, where they're constantly exposed to arsenic or those kind of things. That actually one of the major reason for people having Parkinson's disease, actually, which is another uh, uh, brother, I would say, for the Alzheimer's disease. Like when people work in this field of coal mines, they're very much exposed to this toxic chemicals. So those people can be also likely most get Alzheimer's disease. And yeah, these are the other factors that can also be the reason for people having getting the disease and not.
Okay. So lastly, you are a part of FEA. So can you tell us about the mentoring program at FEA?
Yeah, thank you for asking that. So APA is a very good, I would not say NGO, but it's like a organization back in India. So they generally, what they're doing is a fantastic work for the community. Like they help all the underprivileged students of India provide free education, provide learning English as well, and develop also the soft skills, like for example, how to give interviews, how to write SOPs, cover letters, writing your
Mhm.
CV. So they provide all this education for the kids who belong to a certain class of the society, and also who are very much optimistic about their career, like who wants to do something good in their life.
Okay.
So I remember when I was in Bach Institute in California, like the founder of FEA, his name is Mr. Deepak Chopra. He actually came to Bach, gave a talk about this organization, and
Uh-huh.
then he approached my then PI, Dr. 𝐬𝐭𝐞𝐫𝐯, about this, like whether we can help them in certain ways, because like we are in US, in abroad, obviously there will be a time difference and everything, but he was just discussing, like brainstorming, like what are the different ways we can help these young bright minds of India.
Yeah.
And then I came with a certain idea, like why don't we do, at that time we didn't know about the Zoom and everything, it's like all
Yes.
has taught us about the Zoom, but at that time we used Skype mostly.
Yeah.
And I gave them the idea, like why don't we like bring all the students in one common center, and we can
Yes.
do Skype call to them. In that way, like we can have a huge class of like at least 15 to 20 people, and simultaneously I can talk with all of them, and if they have any particular questions, they can even ping me on WhatsApp separately.
Yes.
Like some students are shy than the other students, right? and then they can talk with me more if they want. And then Mr. Chopra said, yeah, that's a fantastic idea. So that's how I became the first ever mentor of FEA. And now
Okay.
I think we have like over 200 or 250 mentors. If you go to their website, you will see huge list of mentors for FEL. And gradually the word spreaded and a lot of people got involved. And now like a lot of the students are getting helped by us and other people. So I would urge like anyone who is watching this video, just Google FEA. I think they have changed their name recently. It's Freedom Employability Academy, something like
Okay.
that. But just Google, and if you want to help those kids out, just join as a mentor. It will
Yes.
take nothing from you. It will hardly take like 1 hour a month or maybe little more than that.
Yeah.
All you have to do is like just sit down with the students, talk them, just know what they want to do there in their life, and
Yes.
just help in whatever way you can. Like you can help them in building connections, like giving some contact information, or help their English speaking skills, edit their CVs and all. Whatever way you can help them, just help them out. I think that will be very beneficial for the kids. And also, like when we are living, as I said, like I left the country in 2008 when I was hardly 22 or 23 years old.
Yeah.
When you stay so far outside your country, you have that urge in yourself, right? Like we need to do something for the country.
Yeah.
Of course, we can give certain donations when there is a disaster or anything like that. But if you help them on a consistent basis, I think you will also feel good. Like you are also doing something for the country by staying abroad. Of
Yeah.
course, you can go anytime, you can return back anytime and directly work with the students. But if that is not possible, at least sitting from US or Canada in your case, like we can do
Yes.
at least do something to help them out. I think this is one of the best scenario.
Fantastic. Thank you very much for your time today,
Yeah,
Sanjeev.
thank you for having me.
It was great talking to you, and good luck with your research.
Yeah, thank you.